this is isolated from, there is no gene flow actually among this type of this. So this variation actually leave a lot of signature for us that can predict their previous, the situation, as well as it also tell about the current situation, what about this population, whether it will be fit or whether it will be vulnerable or it will be the endangered, like this way. According to this decision, normally we take some, the further step for the conservation. So thank you very much for your kind attention.
So I can't accept question for this because already the time finished. Please discuss in the break session.
Okay, inshallah.
So thank you again for the talk. And now let me invite Mr. Ginanja from the University of Tokyo on next oral talk. And he's going to talk in effects of the PCAR plasmid carriage on the osmotic stress response of the bacteria.
نطلع نطلع احنا الخمسة ولا قبل؟ مشان صار النور يظل.
لا خلص الجلسة.
ايه؟ شو نسوي بوتسكار؟
ما بعرف أنا المرتاحين.
مش مهتمين بس هنا قاعدين.
Thank you so much for the kind introductions. The title of my research is the effect of PK1 plasmid carriage on the osmotic stress response of Pseudomonas bacteria. This is actually my PhD thesis. And when I asked to deliver the presentations to the plurality of the audience, I tried to make it as simple as possible. So, so first, what is plasmid? So plasmid is a mobile genetic element, and it's usually it's a circular DNA like this, and it's independent to the bacterial chromosomal DNA. So usually the total the genome of bacteria consists of two parts: the chromosomal DNA and then the plasmid. So this plasmid also bring another what you call it? Can give another properties to the recipient. For example, ability to degrade pollutants in environments. So my research focused on PK1 plasmid. What is PK1 plasmid? PK1 plasmid is firstly isolated from Pseudomonas resinifrans CA10. It was INC7 plasmid that transferable to another Pseudomonas and Streptococcus bacteria. So what is the meaning of INC7? INC7 is incompatibility. So plasmid group based on the incompatibility, which means that if it's from the same group, it cannot be coexist in the same host. So and then what is the osmotic stress? Maybe you already know, familiar with this topic since high school. So this is the structural of the general structure of bacterial cell. Bacterial cell is prokaryotic cell. It doesn't have endomembrane. So it consists only cell wall, and then cytoplasm inside, and then plasma membrane. And then in the environments, the concentrations of solute or solute is dissolved elements like salt, sodium chloride, or potassium chloride, or whatever. So if the concentration of solute is relatively low than the concentration salt inside of the cytoplasm, the water from the environments will goes into the cell and make the cell burst. And then if the concentrations of solute is higher than the concentration of cytoplasm, it will make the cytoplasm goes out to the cell, to the environments. So that's why the cell needs the osmoregulations to keep the cell condition in the homeostatic conditions, so it can survive. So this previously my my senior analyzed the previously my senior construct a new bacteria which can the new bacteria, the new three Pseudomonas bacteria, Pseudomonas putida KT240, Pseudomonas aeruginosa PA01, and Pseudomonas fluorescens, which contain P-carbon plasmid. So and then this newly constructed bacteria is the phenotype can be measured by these tools. So these tools is biolog phenotype microarray, which can easily measure the phenotype by the cell respiration level. So you can see that this is the cell respirations and this is the time. This is the respiration level of bacteria harboring P-carbon plasmid, and then this is the respiration level of bacteria do not contain P-carbon plasmid, and the yellow one is the overlap. And then what I need to do now is to continue this study into larger scale, which is in the form of bacterial growth by measuring in the optical density 600. So first I will explain about the bacterial growth phase. So bacterial growth phase generally divided into 4 phases: lag phase, exponential phase, stationary phase, and death phase. Lag phase is when the bacteria is preparing for the cell divisions. The exponential phase is when the bacteria actively proliferated or divided. So, and then the stationary phase, and when the bacteria reach the peak, so the number of living cell and the number of death cell is almost equal. And in the death phase the data is not shown. Actually, the growth goes down. Is when the bacteria, the number of cells of bacteria which are alive is less than bacteria is dead. So this is the principle of I measure the bacterial growth by OD monitor C, and it's in the spectrophotometry. So when the light 950 is converted to 600, if the no cell growth, it will be detect 0. But if there's a cell growth, it will be detect, for example, in this case, 0.5. And this is the tool that I use to measure the bacterial growth. And then this is the result of the growth curve. You can see that this is the result of biolog phenotype microarray with and without P-carbon plasmid for KT240, PA01, and PF01. And then it's correlate also with my result. But in this case, what is the most important thing is, interesting thing is, in the 4% KCl, with P-carbon plasmid mostly affect KT240, but not PA01 or PF01. So I'm wondering what happens in in KT240. So I initially want to screen the osmoregulatory genes from PA01. Why I choose PA01? Because it was the most is a pathogenic bacteria which is osmoresistant in human body. So I made a genomic library from PA01. So this is my study objective now. So how I made this is, first I cut I isolate the total DNA and partially digest by enzyme cell3A1, and then I ligate into the vector plasmid. Okay, but I found that from the genomic library, I found that there is a cell that can be growth in the which is good conditions, but unfortunately, when I check, there is no insert. So I tried to screen from the spontaneous mutant, and then from the spontaneous mutant of previously from Pseudomonas aeruginosa. Now I try to screen from KT240 with having picoran plasmid. So I found how I generate spontaneous mutant is only to grow it on the 4% KCl medium, and I picked 30 colonies, and from the 30 colonies I found 6 mutant candidates, and you can see that the candidates is the mutant candidate. So this is control, the blue one and the black one. The blue one is without picoran plasmid, and the black one is with the wild type strain with picoran plasmid. And then the red one is the growth of the mutant. You can see that the mutant growth is always better than the wild type. So I made in 3 replicates in candidate 11, candidate 12, candidate 13, and candidate 14. And then candidate 18. So after I screen, from these 6 mutant strains, I make whole genome sequence analysis, and I found out from the whole genome sequence that the mutation happens in MAXT regions than non-MAXT regions. So this is the type of mutation is mostly single nucleotide SNP, single mutations. So and then after that, I confirmed by RT-PCR, but it's still in progress. Thank you.
Okay, thanks for the talk.
No discussions.
Still, we can accept one short question if you have.
Okay.
Continue the discussion in the break. Sorry for that.
Yeah, it's okay.
And moving to the next, the OT5, let me invite Mr. Hilal Al-Nahyan from Saitama University. And he's going to talk in real-time fluoroscopy simulation of common bile duct stone extraction.
Panjang kan?
Okay, please.
Actually, even though my talk is in the life science field, but actually it's basically from the medical robotics, especially the surgery simulation. So today I'm going to talk about the real-time fluoroscopy simulation of common bile duct stone extraction. So this is the outline of my today's talk. So to understand my talk, actually we need to know what the ERCP means. ERCP is an operation called endoscopic retrograde cholangiopancreatography. So this operation belongs to extracting the stones formed in the liver, gallbladder, or in the common bile duct arena. So, for better understanding, if we look at the anatomy of our human digestive system, we can see that from the mouth there is a long esophagus, and after that there is the stomach, and at the end of the stomach there is an opening, it's called major papilla. In the major papilla, there are 2 openings coming, 2 channels coming, one from the gallbladder, this is the gallbladder, so gallbladder, and another is from the pancreas, which is called pancreatic duct, and this is called common bile duct. So, in the liver, there is generating the bile, so bile stores in the gallbladder, and then it travels through this channel, and the pancreatic juice produced in the pancreas, and then it travels through the pancreatic duct and meet in the through the major papilla in the common bile duct. So in the duodenum, when it comes, it digests the fatty tissues of our food. So what happens, in sometimes there is some accretion of the bile and it generates the stones like this. So if the stones forms in the gallbladder, there is some operation called laparoscopy, and it cut the gallbladder and remove sometimes. But the problem is sometimes the stone form in this tube, or sometimes the small stone travel through this tract, and it resists the flow of the normal flow of the bile, and it creating a lot of unusual behaviors. So what the doctor needs to extract this stone from this duct. So usually to do this procedure, the doctor insert a long endoscope through the mouth, which travels up to this point, but the endoscope cannot penetrate through the major papilla. So the doctor insert another catheter, kind of a stent, inside the endoscope, which travels through this way, like this. And it goes, sometimes it draws the stone, and sometimes it just covers up, wrap up with the balloon and travels through. So with this procedure, it needs around 200 times to get expert in this operation. So what happens if you are the patient and go to the young doctors? So this is really very difficult for the young doctors to operate these procedures, and also for the novice doctors, you cannot get expert until 200 trial. So you cannot have without handling 200 patients. So what is required is, we are developing a training simulator based on the computer programming and the real-time simulation. So in our research, we developed a real-time simulator for the training purpose of this duct. So at the first time, there is no duct visible in this scenario in the X-ray images. So doctor needs to insert some dye, radioactive dye, which makes the duct visible in the X-ray images like this. And when the duct is visible, the stone can be appear, and the doctor send the catheter and to pick up the stone and turn it out. So actually, up to the current simulation world, the market is available with these two simulator, Acutus endoscopic simulator and JMRIS GI method. Both are focusing on traveling the endoscope up to the major papilla, not extracting the stone from the duct. So this was our research objectives. So what we actually did is trying to make a simulation, real-time simulator to train up the young doctors. So there are some previous researches based on the fluoroscopy simulation. Actually, the major challenge of this work is for developing the training simulation is developing the real-time fluoroscopy simulation. Fluoroscopy means X-ray images, real-time X-ray images generation of the total procedure with presenting the global complete deformable characteristics of the models. So these are some previous studies focusing on the fluoroscopy simulation, but those are far away from representing the scenario of the common bile duct. So these are some summaries you can go through as the time is limited. So what we did is we tried to extract the shape from a real patient organ, and there are some previous researchers. They present the size of the duct after getting the dead patient, dead man's organs. So they present some structural scenarios. So we compute those to represent the diameters, and we generate the shape from a patient real fluoroscopy image. Then we extract the shape and generate with the diameters. We generate the shape like this, and then we generate some catheters and also the stones, and make it as a deformable to represent the deformable characteristics with mesh spring systems. And we generate the fluoroscopy for extracting we extract the real background image from the real patient information, and then construct the duct, stone and catheter models and assign a specific attenuation coefficient to represent in the X-ray image, and then combine all the coefficient values to generate the final image. So finally the simulation results like this. It represents, this is the background image we extract from the real patient, and then we generate the duct here and representing the stone here, and send the catheter to travel up to this point, wrap the stone and pick it out. So before going to the comparison, I wish to present a little video. So can you see there is no duct visible here? So from the tip of the catheter, the dye is injected and then gradually the duct is visible, and here there is a stone and then duct goes up. The doctor pushes the catheter through the endoscope. This is the endoscope, and due to the breathing there is a continuous motion of our body, so the total structure is moving. Catheter goes up, and then when it passes through the stone, doctor inflate the baskets, the tip of the catheter, and this basket wraps the stone, and then it gradually pull it out from the. So this is the real procedure, and we simulate it like this. So. So you can see there are a lot of deformation scenarios it needs to represent this procedure. So there are a lot more things to talk about, but as time is limited, I just show some comparison, and in the simulation result we can find out that the computation time is 8 milliseconds, so we can reach up to 30 milliseconds to represent the real-time behavior. So it's far behind, so it's okay, no problem. And there are some, we send this simulation to the ERCP experts, and the experts comments like this: it was a good representation of realistic training simulation, an excellent simulation of contrast agent injection, and the overall simulation was graded as 4 out of 5 in a scale. And in conclusion, we can say it's a quite realistic fluoroscopy image generated in real time, and whereas the computation time is less than 8 milliseconds. So thank you very much, that's all.
Thank you for the interesting talk about the fluoroscopic techniques for the detection. I can accept one short question if you have any. If not, I would like to close this. Thank you
How
again.
many simulation training one young doctor will be expert?
Around 200 trial.
No, on this, also on this system, 200.
Actually, it's possible to, this is for one patient scenario. We can change the scenario with different patient information, but as the computer-based simulation system, this is a part of the computer screen part. There is another device part. So the device will be operated by the doctor, the scenario will be presented, synchronized, and then it will be trained up. So the important advantage is they can practice in their home also to get expert. Without patients.
At least your research will reduce this number of patients in the future for the training purpose, like from 200 to 120, so that you can.
No, this is not done by ourselves. This one research mentioned that to get one operation expert for the ERCP procedure, doctor needs to practice 200 trials.
Right.
So this is the complicated operation procedure. So it needs much more trial. What we are doing is we are making a simulation to make the life easier, rather going for 200 patients, trial in your home for 200 times, and go for the real patient.
Okay.
Thank you very much.
Thank you. So now please note that the OT6 is cancelled. Instead of that, OT12 is going to talk here now. Let me invite Muhammad Rajib from Saitama University, and he's going to talk in the title, Economic Mineral Potentially in Fluvial Sand of Bangladesh. Please talk.
Sorry, this is a sudden change, so I could not copy. But I think I will not take much time. Assalamu alaikum. Well, among all the biological environment, let's move on to some physical environment. I don't know how much I can make you interested about this one. Instead of what I am doing in Saitama University, rather this is my personal research interest, what I was doing in Bangladesh in my office. Mr. Moniruzzaman is one of my colleagues. So I would like to talk about economic mineral potentiality in fluvial sands of Bangladesh. So if some of you know about the geology or geomorphological condition of Bangladesh, it's situated at the foothills of Himalayan mountain range, from where almost all the rivers are originated in Bangladesh. From the Himalayan mountain range, rivers are carrying billions of tons of sediment, and eventually it's going towards the Bay of Bengal. As you know, Bangladesh is almost a flat country, except some hilly region on the southeast part and on the northern part. Almost entire country is a basinal part. Maximum is 10 to 20 meters above mean sea level. So it makes all the rivers coming at a very flat region. So siltation problem, it's quite a big problem, which creates flood hazard and some other social problems also. So what are the placer minerals? If some of you know, it's the accumulation of valuable minerals formed by the gravity separation. The zone of weathering is the source of minerals. By different transport media, it's going towards the basin area, and basically the placers are accumulated due to some gravitational separation technique in the sedimentary process. In beach area, if you go some areas, or sometimes in the river areas, you can see some kind of black patches in the, on the surface. And if we just make a cross section of this type in the subsurface, if the heavy minerals or these black minerals are quite a bit, then you can see this kind of cross section. And just to note that these are pictures. Physically, I have been there, I took these pictures, so these are real, not just from the theoretical one. In placer mineral exploration, Bangladesh Atomic Energy Commission, my organization, has been doing, it's been quite a bit time, around 30-35 years, and we have discovered around 18 heavy mineral deposits along this northern coast and southern coast of Bangladesh. Some of the minerals are something like this: zircon, garnet, rutile, ilmenite, magnetite. All these are, maybe sometimes you heard these names, because these are widely accepted economic minerals all over the world. And some are very nicely shaped, nicely prismatic shapes, like foxylate color, different other colors also. Some of the names you can also know, like the quartz. Yeah, some are magnetic minerals also. These has been done for some time, but unfortunately, not any systematic exploration activities have been done in Bangladesh so far. Some government organizations and some other private companies who are working in some of the parts, except these coastal areas, in the fluvial environments, that means in the river areas, some parts have been explored so far. And recently, what I have tried, along with some of my colleagues, to quantify some of the radioactivity in those heavy minerals or those placer minerals in considerable amount, I would say. For example, around 506 ppm of thorium or 141 ppm of uranium, these were quantified in some fractions of Bangladesh placer minerals. Recently, except these coastal areas, some exploration activities have been shifted because the resource in the coastal area are limited. So where can we get this kind of minerals more? And what we have found, that almost all the rivers, because of the very plain land, the very low gradient, all the rivers are almost filled up with the silts. So the silts could be alternate source of this kind of minerals. So that was my thinking, and based on our primary findings in our, I could say, processing plant, we found that the coastal areas which are very rich in this kind of minerals, some of the places in Bangladesh, in the river